For my midterm, I selected age-related diseases caused by changes in the immune system, and the article I chose to review is called Age-Related Alterations in Meningeal Immunity Drive Impaired Central Nervous System Lymphatic Drainage. The hypothesis behind this research is that age-related dysfunction of the meningeal lymphatic system decreases drainage of the cerebrospinal fluid in the central nervous system, which leads to an increase in the buildup of toxic proteins that cause the plaques in the brain associated with age-related neurodegenerative diseases such as Alzheimer's and Parkinson's. The peripheral lymphatic system has a unique structure, which was not identified in the central nervous system. Therefore, it was largely believed that the central nervous system did not have a lymphatic system. Jensen et al. Described the discovery of cerebral spinal fluid draining into the deep cervical lymph nodes in research published in 2012 by following fluorescent tracers injected into the cerebral spinal fluid. Like the peripheral lymphatic system, the meningeal lymphatics are closely intertwined with veins, in this case, the dural sinuses. Research showing that the lymphatic system deteriorates with age goes back to Pan et al., published in 2008. They examined 161 lymph nodes from 1 fixed and 7 unfixed human cadavers, ranging in age from 81 to 98 years. The nodes were harvested from the cervical lymph nodes. They observed that they had 2 distinct characteristics. They were classified as either solidified or transparent. There were 38 solidified nodes that were palpable and retained the bean shape of a typical node. However, the injectables used to identify the nodes did not pass through them. The follicles in the cortex of the nodes had disappeared, and the medullary tissues had started to degenerate, and some tissues had been replaced with fatty tissues, with less than 50% of lymphoid tissue remaining in these 38 nodes. The remaining 123 nodes were transparent and could only be identified by inflating them with injectables. A cross-section of the nodes revealed degradation in both the cortical and medullary tissues, and that about half the node had filled with fat, pushing what tiny amount remained of the lymphoid tissues over to one side. The lymphatic system originates in the dermis, about 1 millimeter below the epidermis. It's called the lymph capillary plexus or initial lymph capillaries. They're made up of single endothelial cell walls called swing flaps, and a collagen fiber called an anchoring filament is attached to the interstitial fascia and to each cell. It's intertwined with vascular system capillaries. When filtration occurs from the arterial end of the capillary, the fluids fill the interstitium. This increased pressure in the interstitium then pulls the anchoring filament taut, thus opening the swing flaps to vacuum the lymphatic load into the lymph capillaries. The lymphatic load is made up of fluids, cells, biologic pathogens, nutrients, fats, fat-soluble vitamins, cancer cells, cell debris, anything that the body needs transported through it or needs cleaned out of it. It starts at the initial capillary plexus, goes on to precollectors, collectors, trunks, ducts, and the subclavian veins, where the blood pressure is so low that it actually sucks the lymph into it to maintain fluid homeostasis. There are about 600 to 700 lymph nodes, multiple afferent lymph nodes for each one, and one afferent vessel that goes down to the next lymph node. So the lymphatic load actually may be transported through hundreds of lymph nodes before it reaches the subclavian veins. The nodes store lymphocytes, slow lymphatic load to phagocytize invaders and waste, and maintain fluid and protein homeostasis in the body. Justin's team identified vessels that were transporting fluid from the central nervous system into the deep cervical lymph nodes and said, aha, that must be part of the lymphatic system. Noting that they were made of astroglial cells, they became termed glymphatics. Similar to lymphatic vessels, they serve as a macroscopic waste clearance system. They transport metabolites and neurotoxic wastes, including beta amyloid plaques and alpha-synuclein proteins from the central nervous system into the deep cervical lymph nodes. It also transports glucose, lipids, amino acids, and neurotransmitters. Unlike the full lymphatic system, for which skeletal muscles is one of the propulsions, the glymphatic system functions more at time of rest. Alzheimer's and Parkinson diseases are neurodegenerative pathologies commonly associated with aging. Both are associated with the accumulation of misfolded proteins in the brain, such as the beta amyloid plaques in Alzheimer's and the alpha-synuclein proteins in Parkinson's. This is compounded by the failure of the central nervous system to clear these proteins. Rustenhoven noted in earlier research that elevated meningeal interferon gamma cytokines directly impair lymphatic systems and functions, and that hypothesized that neutralization of the interferon gamma cytokines in aged mice may alleviate the age-related meningeal lymphatic dysfunction. This test was done on mice, male or female wild-type mice, in two groups, aged 3 to 4 months and 20 to 24 months. The variable controls included using litter mates whenever possible. The mice were at the facilities and habituated to the climate for 1 week prior to the experiments. They had a 12-hour cycle of light and dark from 7 a.m. to 7 p.m. and they were fed rodent chow and sterile water ad libitum. After their 1 week acclimation period, all mice were anesthetized with ketamine. The back of the head and neck were shaved and cleaned. The incision was made on the back of the neck, and the cisterna magna was exposed. Injections were placed directly into the cisterna magna. Mice either received 2.5 microliters of ovalbumin conjugated to Alexa-Flor 594 or 5 microliters of IgG control antibodies. The younger group of mice received injections of 9-cytomegalovirus to increase their interferon gamma cytokine in the cerebral spinal fluid via the intercisterna magna when they were 8 weeks old. The 20 to 24-month-old mice received additional injections every 72 hours for 8 weeks. Divided into two groups, they received either 2 micrograms of in vivo plus anti-mouse interferon cytokine or in vivo plus rat IgG1 isotype control. After 8 weeks, the mice were then euthanized. Transcardial perfusions were performed to preserve brain and lymphatic tissues. For over 95% of the mice, a single deep cervical lymph node was present on either side of the trachea, and only mice where both deep cervical lymph nodes could be harvested were analyzed to prevent unilateral bias, thus eliminating mice who had only 1 or 3 deep cervical lymph nodes. The lymph nodes were cut to 30 micrometers thick sections and mounted directly onto gelatin-coated glass slides with about 10 to 20 sections per slide spanning the entirety of the lymph node. Therefore, it was the size of the lymph node that determined the number of sections per slide. Each section was analyzed for glymphatic influx and averaged to generate the value for a single mouse. To study the brains and meninges, the mice were decapitated immediately posterior to the occipital bone. Brains were cut and rapidly frozen over dry ice. Coronal cryosections were cut 100 micrometers thick. Single-cell RNA sequencing analysis of the dural meninges was performed on the samples. And the samples included 5 individual young and 5 individual old dural per experiment. There were 2 experiments, the ovalbumin or the IgG antibody injections So there were 10 Dura samples per age in total, making it a total of 20. T cells are major producers of the interferon gamma cytokine. This increases the aging of the lymphatic endothelial cells. Analysis of the mouse dural tissues demonstrated a significant increase in the interferon gamma cytokine protein in the meninges of aged mice compared with their younger group. Aged mice received injections of anti-interferon gamma cytokines, neutralizing antibodies, or IgG isotype control every 72 hours for 8 weeks. Researchers found improved lymphatic drainage of the ovalbumin 594 to the deep cervical lymph nodes with the injections of the anti-interferon gamma cytokine neutralizing antibodies in the older mice. Glymphatic drainage, on the other hand, of the cerebral spinal fluid in younger mice was decreased with the chronically elevated levels of the interferon gamma cytokines. This research shows that age-related meningeal lymphatic decline is reversible, and it highlights the potential for clinical translation of these findings for aged or even late-stage neurodegenerative disease patients. It's exciting to see that there may be medical intervention which may enhance lymphatic drainage. I am a certified lymphedema therapist, meaning I'm trained in complete decongestive therapy, which is the gold standard of treatment for lymphedema and other pathologies of the lymphatic system. Complete decongestive therapy consists of 4 parts. One is exercise. The second is skin care, because the lymphatic system begins just 1 millimeter below the epidermis, and people with a dysfunctional lymphatic system are very prone to infection very easily. The next part is compression to help with drainage. And then, of course, manual lymphatic drainage, a very specialized manual therapy to help to get the fluids moving to reduce the edema and lymphedema by getting the proteins going through the lymphatic system as well. Indocyanine green is being used to trace the flow of lymph. I think it would be very interesting to follow the flow of the meningeal lymphatics with the use of manual lymphatic drainage to see if that could work with and enhance the effects of the anti-interferon gamma cytokine neutralizing antibodies. Perhaps by stimulating the deep lymphatics through the abdominal techniques we employ in manual lymphatic drainage, and then create the vacuum, it may help with the cranial drainage through the glymphatics. Also, there is a specialized technique of manual lymphatic drainage just for the head itself that features techniques that focus on the terminus of the lymphatic system, which is the subclavian veins, and a lot of work on the deep cervical and superficial cervical lymph nodes. It would be very interesting to have some tests done with the indocyanine green to see if that does help with the drainage of the glymphatics of the cerebral spinal fluid from the central nervous system. And finally, here are my references. Thank you.